What's the microscope? Then the microscope obviously is a device that is going to help us to see very small images, right? And that's why we have a microscope. One thing that I want to mention is that there are two main things that you need to know about microscopes. Okay, one thing is parts of the microscope, and another thing is going to be properties of the microscope. Parts of the microscope are the ones that we can see right here, right? We're going to have the ocular lenses, right? Ocular lenses are going to be these ones. We're going to have, this is a lens right here. Okay, and what this lens is going to do is going to magnify whatever that you're going to be looking at. However, you pay attention, there are other lenses right here. This one's right here. This one, and this one, and this one right there. Those are going to be lenses as well. But these ones are going to be called objective lenses. And what are they going to do? The same thing as the previous one. They're going to magnify what you're going to be looking at. So we have two lenses. So then you're going to put whatever that you want to be looking at, you're going to put it right here. And the lenses are going to magnify it for you. Okay, so let me change this color so you can see. So let's say, for example, this is going to be, you want to look at this red thing right here. And then what you're going to do, you're going to put your eyes in here and you're going to see whatever is in there. So then how many times this object is going to be magnified before it actually gets into your eyes? I think it's going to be magnified twice. Right? One time by these lenses and one time by these other lenses. Okay, again, this one's right here. These are going to be the objective lenses. These ones are going to be the ocular lenses right here. This one is fixed. The ocular lens is fixed, meaning this is always going to magnify this 10 times. And this one right here is not fixed. That's why you have different lenses in here. So you have one in here, another one in here, another one in here, another one in here. There are multiple different lenses in here. This part right here rotates. That's why that is called the rotating nose piece. Why you can rotate this? Because that's going to allow you to change the lenses, right? So then you can change this one to this one, or this one to this one, right? By moving the rotating nose piece, right? Which is this part right here. So let's take a look, and let's say you are using this lens right here. This lens is 4. So then how many times you're going to magnify before you actually see that with your eyes? Then it's going to be magnified 40 times. The 4 times right here with the ocular lens, plus times the 10 times right here by the ocular lens. So the objective lens is going to be 4, the ocular lens is going to be 10. So therefore the total magnification is going to be 40. If you use this other lens right here, so let's say, for example, this one is going to be, let's say 40. So then if this lens is 40, then what would be the total magnification? 40 times 10, that would be 400. Okay, and let's say you change this one to this one. Let's say, for example, this one right here. Let's say that one is 10. So then you will change this to 10 right here, right? So then instead of 40, that will be 10. So then what will be the total magnification? Then the total magnification would be 100. You can see right there. Okay, so then the total magnification that you're going to get in your eyes right here varies depending on the lens that you're using right here in the objective lenses. You can move and change the objective lenses by moving the rotating nose piece, which is this part right here. The total magnification that you're going to get depends on the ocular lens that you're going to be using times the objective lens that you have right here, right there. They're going to be located inside there. But that one is fixed. That doesn't change. That's always 10. Okay? And the total magnification that you get again is because you multiply both lenses, and that gives you the total magnification, as you can see right here. Okay, so then going back to this, we have in here the ocular lens. This is the rotating nose piece. These are going to be the objective lenses right here. Okay, and you're going to put the object right here, right there. So for example, this is I'm going to put a slide in here like this, which is a piece of glass, where in there, you're going to find the object that you want to look at right there. Now, this is not in the air. This is obviously it's going to sit in this plate right here. So this plate right here, that is going to be called the stage, which is what you have right here. Okay, the stage is this plate where you actually put the slide so you can actually see it. In addition to that, what other parts you have in this microscope? Well, you're going to have, for example, this, right? This is the arm. This is what you use in order to hold the microscope. In addition to that, using common sense, right? This is going to rotate. Right? You can switch the lenses. And the lenses, let's say, for example, this is magnifies 4 times. This one magnifies 40 times, this one 10 times, etc., etc. So if they have different magnification, but they can be the same size, right? So the one of them is going to be like this, the other one is going to be like that, and the other one is going to be like this, right? Because they're going to have more magnification, right? Let's say 4, this is going to be 10, and this is going to be, let's say 40. We can have another one in here even longer, right? Maybe this is 100. Right? And you're going to get more magnification. So what I'm trying to say with this is, since you have multiple different variations in the lenses, then when you put the slide over here, right, when you get slide and you put it over here, chances are that when you look right here with your eyes, you're going to see nothing. Why? Because this object that you have right here that you want to look at is not going to be aligned with the lens that you have. Right? So in other words, it's not going to be aligned with the lens. So therefore, you have to adjust the position of this object that you have right there in light blue. In order to adjust that, you're going to have different knobs. Okay, so it's not very clear in here because the image doesn't allow you to see that. But right here, you have set of knobs. This one's right here. So this is one set of knobs right there. Right, that is not very clear in the picture because one is in front of the other. And the other knob that you're going to have is this one right here. So you see only part of it right here, right there. And then you have another one, which is this one right there. Okay, so then you have right here, you have two knobs, the top and the bottom. These two knobs right here, at the top and the bottom, these two knobs, what they're going to move is they're going to move the stage back and forth. So to the front and to the back like that. Okay, that's why you're going to use these knobs right here. Okay, so what are these two knobs for? To adjust the stage. They're going to move the stage back and forth. Now, these other two knobs right here in green, you have one big one, you have a little one. So the big one is this one, the little one is this one. What are you going to use these knobs for? These two knobs, you're going to use them to align the stage, to move the stage up and down, like this, and like that, up and down. Now, these two knobs right here, the big one is going to be called the coarse adjustment knob, and the little one is going to be called the fine adjustment knob. The coarse adjustment knob is going to move the stage like this, and like that, fast, and big movements. So you can move the stage up and down very fast. The fine adjustment knob is going to move it like this, slowly. So you can actually see more details like that, slowly, like this. Okay, so you're going to see you're going to be moving that fine adjustment knob very fast, but it's going to move it slowly like this. Coarse adjustment knob just moves it up and down very fast. Okay, so then those are the parts of the microscope. So you have in here again, ocular lens, objective lens, rotating nose piece, plate, knobs. This big one, coarse adjustment knob, fine adjustment knob. This is the arm. This is the light source. Okay, and this is the base. Down here, you have the condenser with the diaphragm in there. Okay, so those are the parts of the microscope. Oh, by the way, we said that these lenses are going to have different values, right? 10, 4, 40, 100, etc. I know it's not that clear in here, but they have a line. This one should have a white line. This one on the back should have a red line. This one right here should have a blue line. And this one right here should have a yellow line right there. And so if it's red, right, that usually magnifies 4 times. The yellow is going to magnify it 10 times. The blue is going to magnify it 40. And the white is going to magnify it 100 times. So this one is going to be the objective lenses. Now remember, the other one is the ocular lens. The ocular lens is the one that is in here, and this one is always 10, right? This doesn't change. Remember, we said parts, and I told don't confuse that with properties, right? What are the properties of the microscope? Properties of the microscope are going to be magnification and resolution. Okay, or let's put in here detail. Okay, so that can help you identify that. Let me show you this now. So imagine that, okay, so you're looking at a microscope and this is what you see. But the image is blurry. Can you see something in there? No. So then what good is a microscope? That it can magnify things, but you can't see it because it's all blurry. The microscope is supposed to make things larger, but if it's blurry, then forget it, right? Then what's the point? Yes, the property of microscope, one of them is to make it larger, but the other property is resolution, which is the ability to see the detail. Right? You're supposed to see the detail. So because of that, then what are the properties of the microscope? They're going to be magnification and resolution. What is magnification? The ability to make things larger, and resolution is the ability to show you the detail. So then right here you have table, low, medium, high, right? Low is 4, medium is 10, high is 40, and oil immersion is 100. So we scroll down right here. Let me see, we have right there, focusing of the microscope. Okay, so in this case, and you're gonna get some slides, and then there you're gonna have a letter E. This is basically for students to begin to practice with a microscope, right? Because imagine that this is the E like that, and the lens is in here. If the lens is pointing down here, and the letter is in here, then how in the world you're gonna see the letter E, right? So then, and this is the case, this is the reason why you use these knobs right here, right? You use these knobs right there. So then when you move this knob, then the letter E is going to start moving over here. Right? So eventually the letter E will show up right here. After students begin to practice with the letter E, and then they get a slide like this. Now they need to identify themselves, right? So then now, this is one cell right here, right there. This is another cell. You can see right like that. This is another cell. We have another one right here. But I want you to see one thing right here. You see the other one is kind of like, if you use your imagination, kind of the other one is kind of like on top of that, right? Because remember, these are cuts of tissues, right? Of group of cells that you have in your body, right? In the person's body. And as you know, right, they are not completely fried, right? They are different levels, right? So as you know, your heart, your liver, those are three-dimensional structures, right? So you can imagine that you have in there cells and structures at different levels, right? So then that's why you also have, you know, something to practice, which is going to be called the depth of field, right? So because remember, you have to understand that if you look at this from top to bottom, some cells are going to be here, other cells are going to be down here, other cells are going to be down here. So you need to understand that, yes, you may see it sort of flat, but it is not, right? Because remember, these are three-dimensional structures, even though when they are cut. So therefore, that's why you're going to have something called depth of field, right? Which is the ability to see that they are actually identified at different levels. And that's what you can see right here. If you look over here, right, then you can clearly see that this is down here, right? And this one is darker, right? Why is darker? Because that one is in superposition of the other one, is on top of the other one. Okay. And obviously at this time is when students start looking at these things and they make drawings of what they see in that particular area.